and he was a native of Charleston, South Carolina. He received his undergraduate and doctoral training in pharmacology at the Medical College of South Carolina, and then went on to obtain his medical degree from Harvard. He remained in Boston for internal medicine and training at Mass General, and then that was followed by 3 years as a clinical associate at the National Heart Institute. He joined Emory faculty in 1964 and then came to the University of Chicago in 1974, and remained as a professor of pharmacology and medicine until his untimely death in 1989. Leon Goldberg was considered one of the giants in clinical pharmacology and was best known for his work with dopamine, having played an essential role in its preclinical and clinical development as a vasopressor. He also can be credited for methyl naltrexone, which was approved in 2008 for the treatment of opioid-induced constipation, having proposed this idea 25 years previous. After his death, there were funds that were put together for this particular lectureship. Another giant in the field of clinical pharmacology is this year's recipient, David Flockhart. He's a professor of medicine, pharmacology, and medicinal genetics at Indiana University. He has a PhD in medical biochemistry from Welsh National School of Medicine in Wales, UK, and then an MD from the University of Miami in Florida. He is the Harry and Edith Gladstein Chair in Cancer Genomics and Director of Clinical Pharmacology at the Indiana University, as well as Chair of Indiana Institute of Personalized Medicine. As a physician, Dave has spent the last 20 years of his career focused in the area of valuable pharmacogenomic markers in the fields of neuropsychiatry, cardiology, and cancer. He was the founding director of the Indiana Institute of Personalized Medicine and supervises over 40 faculty. Since 2011, he has also served as the Associate Director for Therapeutics of the Indiana Clinical and Translational Science Institute, a role which coordinates research at Purdue University, Indiana University, and Notre Dame University. He has been continuously funded since 1993. He's had an illustrious career getting into pharmacogenomics very early. Sorry about this. And was one of the first to identify that pimozide, which is an agent used for Tourette's, was metabolized by CYP2D6, which is not present in about 7% of Caucasians. And this was a very important work understanding the metabolism of this particular drug. One of his most impactful accomplishments was in the field of tamoxifen and breast cancer, identifying that CYP2D6 was also important in the metabolism of tamoxifen to endoxifen, a highly active metabolite. And this data was very important not only in understanding its metabolism, but also to get a better idea of drug-drug interactions. He was able to identify that the combination of SSRIs with tamoxifen could compromise the care or the outcomes of patients. And these SSRI drugs were used for the treatment of depression and hot flashes, but they blocked the activity of CYP2D6, and therefore there was less active tamoxifen or endoxifen present in the system. So Dave is currently the co-PI with Dr. Paul Dexter of the Genomics Medicine Demonstration Project termed Ingenious. This is all part of the Ignite network that is funded through the National Institute of Health. And in his particular trial, he's looking at evaluating 6,000 patients in the Indiana area, both at Indiana University and also at community hospitals, and evaluating the economic cost of bringing in pharmacogenomic tests over a period of 1 year. So on a personal note, Dave is one of my all-time favorite colleagues. No pretenses, just one of these guys who you want to have a beer with at a meeting, or maybe two. And in September 2014, Dave was diagnosed with glioblastoma multiforme, and he had an initial response following radiation and chemotherapy, but then he recurred in May of 2015. He then went on a clinical trial here at the University of Chicago and is now dealing with some dysphagia and is unable to communicate. So we have his fellow here, Dr. Namita Dave, who's going to present his work. And I just want to say as a side that Dave has also put on a number of interviews on NPR, which are just fabulous. It really speaks to his courage and fortitude during this very tough time in his life. So I'm just going to, we're not going to show these now, we'll show one at the end. But these are a number of, they're about 9 or 10-minute interviews talking about just not only the frustrations, but also the challenges of being both a physician and a patient. And some of the trying to get back through rehabilitation, things that we all take for granted, like texting and so forth. And so I'm going to present Dave with the award here, and then we'll have Namita give his talk. So Dave, a giant... You can go sit down. Oops. Yeah. And now we'll have Namita give his presentation.
So on behalf of Dave, thank you, Eileen, and the committee for giving him this honorable award. And it's an honor for me to talk about his work and his vision for the future. So the first part of the talk is going to be about his adventures with outliers, what led to his entire career over the past 20 years in pharmacogenomics. And after that, the last part of the talk is mainly his message to all of us about the future of medicine. So the very first adventure that Dave had as a resident was with clomipramine. So there was a 17-year-old female with lethargy and palpitations. Her condition was that she had 2 weeks of palpitations, no syncope, no leg pains, no intolerance, and her history was OCD for 3 years. She was treated with fluoxetine for 2 years, but had persistent insomnia, and so was changed to clomipramine about 1 month ago. Now, here is the patient, and that's Dave back in mid-1990s. And so what he did is he did a small experiment with himself serving as the control and the patient. So he did a single-dose PK study to look at, compare the PK profile in himself against the patient. And what he saw is that in the patient, the half-life of the drug was 3 times longer, about 120 hours. And this is where he did some Western blots looking at the gene's DNA and found out that CYP2D6, this patient had both of the variant homozygous, and that's why there was such high concentrations of the drug. The next adventure that he had, this was, I think, 1998. So there was a 27-year-old patient with Tourette's who was switched from haloperidol to pemazide due to persistent fatigue. And the ECG intervals were normal before treatment. Gradually, they increased the dose of pemazimide to 14 mg per day, while the normal usual dose is 1 to 2 milligrams per day. And he was presented to ER with bronchopneumonia, for which he was prescribed clarithromycin 500 milligrams twice a day. 4 days later, he had syncope and was taken to ER. His pulse was 63, QTC 506, and instead of observing him for a long time, some time, he was discharged to home. The next morning, he was found cold and unresponsive, and he was pronounced dead on his arrival to the ER. Now, at autopsy, they looked at the concentrations of the drugs, and they found that pemazidine was 50 nanograms per ml, while the usual concentration is 15 to 20 nanograms for the dose given. So this led to Dave and his group looking at Did pemazide prolong the QT prolongation, or was it clarithromycin? Now, as we all know, erythromycin is known to prolong QT interval. So the very first thing the hypothesis was that probably it is clarithromycin. Now, Desta, who's now a professor in our division of clinical pharmacology at IU and an interim chair of our division right now, he was Dave's fellow back in 98-99, and he did this clinical trial. So what they did is the placebo group is the one where they got just pemazide and then co-administered with clarithromycin. What they observed is on co-administration of clarithromycin, the concentrations of pemazide increased, and as well as those were the patients who had QTc interval prolongation. So it was now in the currently, if you look at the label, you'll find that it says that it's metabolized by CYP3A4 and should not be concomitantly administered with inhibitors of this system. So clarithromycin was the inhibitor which was causing this drug-drug interaction. So this is his group, Georgetown lab. Dave was very particular that I take names of all the people, and that was been the hardest part for me because I don't know all of them personally, but this was his graduate student, Milind and Jimmy. That's Mehul Desai, who was a fellow. Nadia, that's Joseph. That's Alex and June. That's Desta at the back, who's still working with us. And Jose, who's now a faculty in Brazil, and a lot of his students come as fellows to our department. And that's Sean, that's Caddy, and that's Dave. I don't think he's changed much since then. So his third adventure, tamoxifen, which went on for a long time. The story, so basically, as we all know, there are several different FDA indications of tamoxifen over the years from 1977 to 2000s. And the most important one is the adjuvant post-menopausal node-positive cancers, breast cancers. So tamoxifen has reduced recurrence of breast cancer in 45 to 50% in adjuvant setting, and it's also reduced the risk of breast cancer in high-risk women by 50%. So there was a case that came to Dave that 45-year-old woman presented with intense, intolerable hot flashes when prescribed tamoxifen 20 milligrams per day, and when she was placed on antidepressant paroxetine, there was resolution of these hot flashes within a week. However, when she was taken off paroxetine, the hot flashes resumed. So this is where what Eileen already talked about, the drug-drug interaction of tamoxifen with antidepressants, that work began. What Dave and his group saw, this is the HPLC chromatogram, where tamoxifen is administered alone, and then this one at the bottom is when it's administered along with paroxetine. So what they observed is there was this one peak of one metabolite that got really low after administration of paroxetine. And they separated, purified, identified, and synthesized this metabolite, and that is the metabolite endoxifen. So what they now, I'm sure clinical pharmacologists have seen this, but we have it on our website. This is the card that Dave has prepared for all the make it easy for physicians and clinicians to use this while administering drugs. And you can see that paroxetine is a CYP2D6 inhibitor. So he worked a lot more on this to show that it was actually, endoxifen metabolite is mainly through CYP2D6 metabolism, and the other main metabolite is 4-hydroxytamoxifen, which is mainly through CYP3A4. And endoxifen is later metabolized by UGT2B7. So he also looked at concentrations of both the metabolites before and without, this is where only tamoxifen without co-administration of paroxetine, and this is after co-administration of paroxetine. So what they saw was there was no change in the concentrations of 4-hydroxytamoxifen, whereas concentrations of endoxifen reduced to a huge level in most of the patients. But they also observed that there were some subjects where this change in concentrations wasn't observed. I mean, to begin with, they had really low concentrations of endoxifen. And so these are the subjects where they found that C2D6, they were poor metabolizers. So they had lower levels of endoxifen to begin with. So this was his group at IU when he joined became the director of clinical pharmacology there, the chair. And some of his people joined him, moved here from Georgetown, including Millian, Jimmy, Desta. He recruited Todd, who was also at Georgetown with him here, Tom Callahan, and some of his fellows and students. That's Jamie Renbacher, who was his fellow that time, and she's currently a faculty. And that's David Jones, and he is now the director of our analytical core. There's Lang Lee, who's our main biostatistician. And this is Stephen Hall, who is now at Lilly. And this is Ray, who's a faculty at Purdue. So hopefully I've covered the key people. And this was another paper where they looked at effect of metabolism inhibitors on CYP2D6 genotypes too. So this graph is for a homozygous wild type variant, and this is homozygous and heterozygous wild type and variant genotype. And then poor metabolizers are the ones with homozygous variant genotype. And these are the levels of endoxifen concentrations in the absence of CYP2D6 inhibitors. And then these are the levels here. So there's a reduction again in the presence of CYP2D6 inhibitor. Now, he also looked at different SSRIs and their effects. So venlafaxine is a weak CYP2D6 inhibitor. Sertraline is a moderate CYP2D6 inhibitor, and these are the strong, more potent CYP2D6 inhibitors. So again, just to show that it was mainly CYP2D6 that's involved in the formation of endoxifen. So a lot of this group, sorry, I forgot to mention, you see this COBRA. So that group has done a lot of this work, and some of these people, this started when he was at Georgetown. So that's him, Todd, and Desta from Georgetown. And this is Michael Johnson, who did a lot of the pharmacology work, and he's still at Georgetown. This is Dr. Daniel Hayes from University of Michigan. He heads the breast cancer oncology program there. She's Dr. Werde Stearns from Johns Hopkins. And this is Anna Mara at IU. So this COBRA consortium, this started when he was at Georgetown, but it continued on when he went to IU. So he expanded to include even the patients at IU Medical Hospital. So a lot of this work was done there. And in 2005, 2009, Todd and Desta also looked at the data of all these women who had got tamoxifen. And what they observed is, even in this huge database, that patients who were co-administered any antidepressants, they seem to have less of cancer-free survival. So this was to show the clinical impact of what what started as a case report, and then moving on to clinical trials, and then also looking at the database available. And recently, one of his graduate students, Jessie Liu, she also looked at all the metabolites of tamoxifen and their effect on aromatase. And what they have shown in this paper is that some of the metabolites of tamoxifen are also aromatase inhibitors, especially here the endoxifen. And the most recent trial that he moved on to look at aromatase inhibitors, pharmacogenomics, but they don't seem to play a huge role. But this was one of the big trials, and the analysis of this data is still ongoing. So they enrolled 500 patients, and they were given either exemestane and letrozole. So this was to compare between two aromatase inhibitors. So what they did is, out of 368 subjects, 211 subjects received one of the aromatase inhibitor. And if they had any symptoms or side effects such as hot flashes or musculoskeletal issues, they were crossed over to second aromatase inhibitor. And what they observed is that about 40% of patients seem to continue the second aromatase inhibitor therapy after they are crossed over. So they don't know what the reasons are. They couldn't find from the study the absolute predictor, but they are still trying to do a lot of covariate analysis right now to figure out, get more data. So mainly, CYP2D6 testing is likely to play a valuable role in some patients. And endoxifen is currently in human testing by NCI, and also 2 commercial companies are working on it. So that was his big contribution to the field. So this talk, he wants to conclude that outliers matter, science is an adventure that matters, and most importantly, people matter as much as data. And here are his main mentors that he's really thankful to. Dr. Abernathy was his mentor at Georgetown during his fellowship. Craig Bretter is one who recruited him at IU. And Ray Woosley was also at Georgetown. And from the stories he's told me over the time, I've known Dave only for about over a year. And from all the stories that I've heard that Dr. Abernathy was really critical of most of his ideas. When Dave used to go over to him and he was like, oh no, that's not gonna work, that's not gonna work. And Dave thinks that really helped him. So his point is, at the beginning of your career, you really need to have mentors that can critically evaluate your work. And this was the part he really wanted me to talk about, and this is his message to us, that personalized medicine will be jump. Now, why should we jump? That's because the opportunity outweighs the risks. We have seen many safety disasters in the past, but after every disaster, we again have jumped, and that's how we have made progress in the field. Now, who's this we? It includes all of us, the patients, nurses, physicians, FDA, CDC, and even the medical insurance companies. So we all need to come together to make this jump. The reasons that Dave is really frustrated of hearing is, okay, it will cost too much to gain too little. Money needs to be diverted to more important things, such as prevention of disease or treatment of malaria. And also that some people say that we are not yet technically ready, the I can't swim syndrome, according to him. And for example, he just wants to show that this is Wishart Hospital, which as of last year does not exist anymore. It has been incorporated into the IU Health. But this was where Dave worked, and this is where the clinical pharmacology division MDs work and used to. So his point here is that this is in 2013, the first prescriptions of drugs where pharmacogenetics play a role. So that's such a huge number of drugs that are prescribed where it's important to know the pharmacogenetics. And we still think that we still are giving reasons not to use this. So one of the review articles he has from a lot of data they had gathered from randomized controlled trials that shows that if you look at this is the benefit versus harm, and about only 10% of patients actually subjects have large benefit without any harm. About 10% have huge just harm and no benefit at all. And about 30% lie in this area where there is mixed benefit and harm, and almost 50% have large, neither harm nor benefit. So this is to show that this one size fits all is not working. And the rationale is that we need this to, we all have not been persuasive enough. The other thing is it's ripe for translation into practice. It just needs a financial rationale. And the pharmacogenomic testing can reduce healthcare systems. And we all know that the healthcare system is under huge financial stress right now. So how can personalized medicine reduce costs? And this is what also led to, I'll show in a few slides about the ingenious study that Eileen also talked about. So this was the main reason why he went into that direction now. So at IU Clinical Pharmacology, the lab, to look at your pharmacogenomics, they look at about 50 different variants of 18 genes on one chip. So the test costs only $250, and that can cover up the pharmacogenomics for all these drugs. So they have worked on his point is that if we try to do a test where we can look at multiple genes, and that can also reduce the cost, and it can be used if you keep it in the patient's records. If he's going to be prescribed on any drug later on and has any of these variants, that can help. So his idea is to integrate personalized medicine into large healthcare system and continuously measure cost of the system and compare them to a non-tested population. And this is the data we should be using for scientific discovery, management discovery, adaptive designing, and rapid dissemination. So this is what led to the ingenious study, which is Indiana Genomics Implementation, an opportunity for the underserved. What the idea is, so Eskenazi Health, a public healthcare system which is focused on treating underserved population, which is right in downtown of Indianapolis, right behind the College of Medicine. And in IU Health, we have 18 hospitals and 56 clinics across the state. So his idea was to reach out to all these hospitals, clinics, and try to implement this. And the investigators on this trial are Paul Dexter and Dave Flockhart. And the co-investigators include Anne, Desta, Todd, Brian, Mark, Victoria, Ken, and Janet. And Victoria Pratt is the director of our Pharmacogenomics Center, core. So this is a collaboration between all these different institutes and also the Indiana Institute of Personalized Medicine, which Dave founded in 2012. The study scope was to have about a total of 6,000 patients. So 2,000 patients in the study arm and 4,000 patients in the control arm. And the aims were to study the hypothesis that CLI certified genotyping targeted at 24 widely used drugs is associated with significant reductions in hospital and outpatient costs over 1 year, and also to test whether pharmacogenetic testing is associated with significant improvement in clinical outcomes of these patients. In order to implement this, we needed IT innovation, and this company, Regenstrief Institute, will have more than 40 years of working with medical records. So they are the ones who are helping with all the IT stuff. So the way this study is going on is that we have this list of drugs. Based on the number of prescriptions prescribed, we have narrowed down about 24 drugs to look at. And whenever a patient is being prescribed that drug, on your screen, there will be a message popping up about the ingenious trial and asking would you like to get enrolled in the trial. And so the physician asks the patient. If the patient says yes, they quickly page the research nurse, and she will come over, do the informed consent, collect a sample, send it to the analytical pharmacogenomic core. And once we have the report will be put into all the pharmacogenomic results will be added to the medical records. But if the drug that she's being prescribed and there is an actionable gene, then there will be a message sent to the physician that action needed. And in that case, it's up to the physician. They'll also have a message popping up, need consult or no. So if they want one of the clinical pharmacologists to go over and consult the patient, that's available too. And if they choose to adjust the dose or change the drug, they add in that information there. And so this is what IT looks at, all the data that they gather. So the plan is to look at cost analysis, and the costs will be evaluated based on empirical data wherever available, as well as some estimations. And the empirical data is based on all the centers for emergency, outpatient, and inpatient costs. And healthcare cost in each arm will be analyzed. So this is what is the projected care cost savings. That about average cost per patient is about $9,200, and 2,000 subjects in this genotype study arm. So the total cost is about $18,400,000. Now, even if we reduce just 10% cost in these patients, that's projected savings of $1,800,000. And so these are the people at the Institute of Personalized Medicine, and this is the reason he wants me to, the thing he wants me to point out here is the diversity we have in this group. That we have people who are OBGYN fellows, OBGYN doctors and faculty. We also have ped, hemonc, and also just basic scientists like myself and physicians and all of them. So here are some of David Haas and Sarah Quinney. She's also clinical pharmacology faculty, but also works in OBGYN area. David Haas is an OBGYN physician and also a faculty at ClinPharm. That's Todd Scar. And we also have a very strong modeling group. That's Robert Bees. That's Michael Eden, which many of you would know. He was a fellow here and now a faculty there. And that according to Dave, is one of the superstars. So he is he's in nephrology and also a joint faculty in clinical pharmacology. And on this side, we have Ken Levy. This was Dave's last PhD student, Jin Liu, who is now at FDA. Then Brian Schneider, who's a breast cancer oncologist. And this is our wonderful staff, Christine, Cornelia, Passion, and they help us coordinate everything in the office. And then we have Tom Callahan, Beth, and some of the OBGYN fellows and the PED fellows, and also the T32 ClinPharm fellows. So this is his family, work family. So we need to jump because there's opportunity to reduce adverse drug interactions and also an opportunity to improve efficacy. And as we pointed out, probably an opportunity to also decrease costs. Now, what do we need to do this? What about the workforce? So this is a graph which shows these are all the total number of MDs, physicians, and 82% of them are involved in non-research, non-clinical practice. About 1% are involved in non-research patient care. 2% are basically, I don't know what unemployed means, but MDs, and then 12% are in academia. So only 12% we have here. So what Dave's point is that what we need to do is basically retool it. We need more of these physicians to be involved in research. So, and we also need to have more money in this field. So how do we attract creative private investment? So right now, most of this is going to IT and green energy investment, which is also, because there is more money there, they are also attracting the best and the brightest. So we need to sell clinical research as attractive to young people and also to venture capitalists and to get more investment in the field. For example, if you look at the IT or this chip, from 128 MB to 128 GB, we made this jump in just about 9 years. We need to make these kind of jumps in our field of biomedical research. And according to him, the future of therapeutic monitoring is looking great. We have more and more of wearable devices and real-time data collection. Dave is a big fan of these wearable devices, counting his steps, looking at his temperatures and everything. I think he got the Apple Watch the very first day it was launched. So according to him, it's looking really bright. And this was his, one of the trips we were making to University of Chicago, and this was his statement that I wish I could live up to 150 years because the future looks so bright to me. So. And so he would like to conclude that, yes, we all need to get together and jump because the opportunity outweighs the risks. And we can jump with people trained enough in diverse fields. And we also need more medical school teaching, which emphasizes on outliers and looking at personalized medicine and aggressively court VC for research and court the best and the brightest. So with that, he wanted me to conclude the talk with this slide of future innovators who, according to him, are in his group. Is Michael Eden, who's a faculty now, and he just got his K award. I swear Dave asked me to put this here. And Kim, she's a wonderful Todd Scars graduate student, and she's already got tons of awards. So he thinks we all are, so we have a lot to prove today. And with that, thank you all. So since we have some time, we also had a video of one of the NPR talks. So he gave 5 different talks during his entire journey over the past year as a cancer patient and a scientist or a physician. And this was the 5th talk, which was in February of 2015.
Dr. Flockhart has been a frequent guest on Sound Medicine over the years, explaining issues in his field of pharmacology. After both chemotherapy and radiation, he's in the midst of a 20-day break from treatments. He told me that, not surprisingly, they really took it out of him.
But the trick that's always kept me going is 2 things. One is if I get up and get moving a little bit, then that gets me over this hump. So I've been able to not maintain my energy levels. My stamina is not as great as it was, but I've managed to understand how far I can go and be firm about when I can't go.
Another Sound Medicine regular is bioethicist Eric Meslin. He's known Dave since they first met while at Georgetown University. Well, they've been kindred spirits ever since a lunch meeting.
Where, I kid you not, we pulled out a napkin and started to draw some ideas about collaborative programs in pharmacogenetics and ethics.
Eric gave Dave a copy of a paper about cancer survival, written by Stephen Jay Gould.
Gould was a famous evolutionary biologist at Harvard University, a very gifted science communicator, and he wrote this paper called "The Median Isn't the Message." And it recounts his own tale of being diagnosed with abdominal mesothelioma and then learning that the median survival in terms of years was a very small number. It was about 8 or 10 months. And it was a wonderful explanation for a non-statistician like me about what median means. It means 50% of the people will live to that period, 8 months or 9 months or whatever the number is, but 50% will live longer. So he describes the story of picking himself off the floor at the Harvard Medical School library when he learned about this wretched disease, but that there was a sense of optimism in what are the chances of me being in the 50% that might live more than 8 months, perhaps 10, 12, 15, or 20 months. And I found it an interesting paper just to learn about statistics, and Gould was one of my heroes, one of my idols. And I wanted to share one of my favorite authors with him. It was almost like a mini book club.
Everybody thinks that the middle statistic is the statistic. Mm-hmm. It's the one that matters. You're going to live 2 weeks. You're going to live 10 years. That's the number I'm going to live. That's the number that psychologically gets into a patient's head. But what is really hard to understand sometimes is that there's a high percentage of people who live a very short time, and that's surprising when it happens and scary. And there's a high percentage of people who also live a long time. But in the middle, we call it the middle statistic. So, for example, in this disease, the disease that I have, glioblastoma multiforme, there are people out there who live 14 years. That's really different from someone who's going to live 12, 14 months, which is the middle. It's also different from the shorter one as well. It really gives one a perspective on the need to think about it day to day rather than be waiting for 14 months to come and then here I am, I'm ready to go. It's a little bit more complicated than that. But I think for patients, that's a tremendously important thing because there are things you can do that put you on the longer side of the spectrum than the other.
Doctors Flockhart and Meslin have taken their collaboration on the road, doing a medical grand rounds in Florida on the issue of evidence-based medicine.
One of the things that I have found useful is really understanding the evidence. When it looks like there's no evidence, okay, go back and look and look and look and look and look, and then really look to see if there was evidence that you had missed in your early literature searches. And when you don't have evidence, one is very prone to bias.
As part of his research into his type of brain cancer, Dave Flockhart turned up an unexpected bit of family history.
My mother had had the same disease, down to the same histology. So this was very, very, very interesting to me, and we embarked on a family exercise to collect everybody's DNA, about 12 of us, I think, to have it all analyzed and see who's got it. And we haven't got through all that yet. But it really stimulated a new thing just because I started thinking about it. And I could talk about it a lot more specifically in relation to our family, but this is not thought of as a genetic disease.
The lifetime risk for brain cancer is 0.6%. Lung and breast cancers pose considerably higher risks.
So to have 3 people in the same family seemed pretty remarkable to us, and that set off a whole new thing that I certainly had not anticipated beforehand. But the minute you bring genetics up, you're back with really interesting questions. If you had it, would you want to know? Even already within the family, there's the question of, if I had it, would I really want to know?
And you have 3 children.
Yep.
Have you broached the subject? Are they getting tested? Or
They've been.
They've been tested. Okay.
So, sorry, we have the DNA.
Okay.
We haven't got the data yet.
Okay.
Right.
So now they're going to have to decide.
Tension in the story here.
Yes, exactly. And have they decided whether?
No.
But at this point, there's no genetic marker for the cancer that David Flockhart has. So the plan is to take those DNA samples from his family members and analyze them, looking for possible patterns.
So this is where bioethics and genetics meet, not in the theoretical classroom, but right smack in the middle of a family.
That's right.
I mean, there's information. There's a little piece of material sitting in a laboratory that the answer's already there. It's just whether you want to open the envelope and find out.
Does this help? I mean, in terms of when you're living with cancer and you're living with uncertainty, I mean, that ability to say, hey, we can discover things. I mean, I'm not talking about whether your children or not want to find this, but the act of science, the act of moving science forward.
Yeah. No, that's hugely important, at least to me. Part of what I want to do is bring more rationality to the public discussion of cancer and to the discussion of science in general. But the whole raison d'etre for the discussion, if you like, is partly because I approach it with a sense of wonderment and a sense of wow and a sense of isn't that interesting? And that's just as potent a motivation for me. It's more potent, actually, as it might be to sit there and wonder why the sky is blue or why this happened to me this way.
But you've always been like that.
I'm just weird, you think?
You've always been interested in science with that sense of wonder. I don't think anybody can, especially in the fields that you've been working in, this intersection of genetics and pharmacology, and why is it this way?
And the most fun thing is when you get an unexpected result out of that.
That's Dr. David Flockhart of the IU School of Medicine, along with his friend and colleague, Dr. Eric Meslin. We'll keep you posted.